Okay, so hello and good evening to everyone. It's 8 p.m. sharp. At first, I would like to thank you everyone for your participation and your willingness to support our program. And to make a brief self-intro about myself, I'm actually one of the Academics Executive Committee for KMS, as well as the President for KMS Power Program. And my name is Ho John Wing in full, or just call me as John in short, as you guys can see down at the name down there, okay? So regarding our event this time, it's basically known as KMS Open Online PAL Session, or in BM, you guys can just call it as Sesi PAL Online Terbuka KMS. Okay, so basically, this is a program being held by KMS. And the main, as well as the prime objective for our event this time would be to provide a chance for all the students in Malaysia, for all the matriculation students in Malaysia especially, that are not just from KMS as well as from other colleges, to experience and get engaged in our KMS PAL program. Of course, PAL program is being carried out in other colleges as well, but we just wish that everyone can experience from the aspect of KMS. Okay, and of course, this is also a brief preparation for all the matriculation students to face your coming PSPM1, which is just around the corner, if I'm not mistaken. Yeah, and so hopefully by the end of our program every day, all the audiences and participants can learn something useful from us, from the power leaders that we have currently in KMS. And so without wasting much of our time, I'd like to go straight into our first day of our program. So let's give a round of applause to our power leaders in our first day of power session sharing, who are Amira, Nicholas, Seema, and Tang. Okay, if possible, I would like to have our power leaders to explain themselves, to make a self-introduction so that everyone in the stream can know them well. Okay? Yeah.
Okay. So hi everyone. I'm Nur Amira Binti Muhammad Medina, and I'm from College Matriculasi Selangor. So I'm one of the biology PAL leader for KMS.
Hi everyone, I'm Seema. I'm also from KMS, and I'm just a part leader, and I'll be presenting chapter 8 today.
Yeah, good evening, everyone. My name is Nicholas Tang Kai. I'm from College Matriculasi Selangor, and I'm also a PAL leader for biology. And later today, I'll be presenting chapter 9. Yeah, that's all.
Hi everyone, my name is Quan. I'm one of the power leader in College Matriculasi Selangor. Today I'll be presenting about chapter 9.3.
Okay, so basically, four of them will be the power leaders for our program today. And to make ourselves clear, Amira and Nicholas are actually the heads of biology subject in KMS, and Pang and as well as Seema will be the assistants for them for today. Okay, so apparently, without wasting much of your time, I would like to have Amira to start her presentation for today. And of course, for everyone's information, our program for today will be as long as around 2 hours, duration of 2 hours. This whole 2 hours will be about biology. So right now, currently, I would like to have Amira to present her slides and also to discuss the things that she wishes to share with everyone of you. Okay, Amira, just go ahead.
Okay. Okay. So hi everyone. Today I'm gonna. I hope all of you are fine at home. So today I'm gonna revise about chapter 7, which is mutation. So I hope all of you can see my slide. Okay. So for the first start, okay. So mutation. The only thing we should know about mutation is all about the classes of mutation is gene mutation and chromosome mutation. So we just need to know about mutation is the classes of mutation. First is gene or point mutation. So gene mutation is a change in a gene at a single nucleotide pair, and chromosome mutation is a change in a gene, but the alteration is at chromosome, not at the gene. So for gene mutation, the alteration is at the gene, while chromosome chromosomal mutation, the alteration is at the chromosome. So we're going to go through this chapter really briefly. Okay. Okay, so in the gene or point mutation, there are 4 types of gene mutation. Okay, firstly is the base substitution. So base substitution is a replacement of one nucleotide and its partner with another pair of nucleotides. So in the example, as you can see, okay, all that we know is T will pair with A and G will pair with C. So here, the T, if we change it into G, so the pair of T, which is A, will also change into C because G can only be paired with C. Okay, so for second, for base inversion, so 2 base pairs or more are inverted in nucleotide sequence. So if we change the C and T, we just inverse it. Okay, from C to T, we become T to C. So the partner will also change. Will also change their position. Okay, so for the third part is base deletion. So base deletion and base insertion. So deletion is for loss of nucleotide, while insertion is addition of nucleotide. So for deletion, as you can see in the example, the base pair TA is missing for the second part after it undergoes the gene mutation. So for base insertion, is previously it have CT and then it becomes CGT. It just will add a base pair GC. Okay. That is all for gene mutation. Now we'll move to chromosomal mutation. So this is the overview of chromosomal mutation that you have to know. And if you deeply understand each part of this, then you will absolutely can conquer this chapter. Okay, first we will go to changes in chromosomal structure, we can call chromosomal aberration. So for chromosomal aberration, there are 4 types, which is translocation, duplication, deletion, and inversion. Okay. So for deletion, okay, deletion is actually we remove a segment of the chromosome. Okay, so as you can see here, we remove the segment B. So after it undergoes the chromosomal mutation, which is chromosomal aberration, so it becomes ACDEF. Okay, so that is for deletion. And then for duplication, so duplication is when we duplicate or we repeat a segment of chromosome. Okay, and then as you can see and in the example ABC, but after it undergoes the chromosomal mutation, it becomes ABBC. So we repeat the segment of B. Okay, so that is for duplication. Now for inversion. Okay, inversion is a segment of chromosome is turned around 180-degree within a chromosome. So there are 2 types of inversion. First is pericentric and second is paracentric. So you can see in the example for pericentric and paracentric, the difference is pericentric will include the centromere, while paracentric will not include the centromere. Okay, so the blue line is a centromere. So as you can see, DEF, it will become FED. It will invert, it will turn around 180-degree and it become FED. Okay, that is for pericentric. It includes the centromere, which is the blue line. So paracentric, it does not include the centromere. Okay, because the centromere is here. So in this example, we see that it does not include the centromere. Okay, so the last one for chromosomal aberration is translocation. Okay, so translocation is actually the movement of a part of the chromosome, to another part of the genome. Okay, so actually translocation, there are 2 types of translocation, which is non-reciprocal translocation and reciprocal translocation. But I only include the non-reciprocal translocation here because the non-reciprocal, it have 2 types, which is intra-chromosomal translocation and inter-chromosomal translocation. So as you can see here, intra-chromosomal location, it involves in the same chromosome. So B and C will change its position in the same chromosome. But for inter-chromosomal translocation, it will change its position into another non-homologous chromosome. So it will change to another non-homologous chromosome, not in the same chromosome. So that is the only difference between intra-chromosomal and inter-chromosomal translocation. Okay, so next, and that is all for chromosomal aberration. So now we'll move to the chromosomal number. Okay, so this is something that you have to take point of. Okay, so for changes in chromosomal number, there are two types, which is aneuploidy and polyploidy. So aneuploidy is addition or losing one or more individual chromosome because of non-disjunction. Okay, non-disjunction is um, really an important part in this chromosomal changes in chromosomal number. Okay, what is non-disjunction? Non-disjunction is a condition where homologous chromosome fail to segregate during meiosis 2, meiosis 1 or meiosis 2. Okay, so in the aneuploidy, there are two conditions, which is first non-disjunction of homologous chromosome in meiosis 1. Okay, and the second one is non-disjunction of sister chromatid in meiosis 2. Okay, so let me explain to you. Okay, when the non-disjunction occur at the meiosis 1, the chromosome, homologous chromosome fail to segregate, right? Okay, so the pair of it will come to this part. We will produce at this part, and then as you can see, this part, it have no chromosome. It lack in one chromosome. Okay, so the gamete produced will become n+1 and n+1 because it have an addition of another chromosome, which is the another side of the gamete will have n-1, which is it lack in one chromosome. Okay, that is for the non-disjunction of homologous chromosome in meiosis 1. Okay, for non-disjunction of sister chromatid in meiosis 2. Okay, as you can see here, in meiosis 1, it have normal meiosis, and for meiosis 2, the non-disjunction occur. So the homologous chromosome fail to segregate. So here, there are no non-disjunction occur. So the gamete produced is N, which is a normal gamete. Which but this part, one of it will have an addition of chromosome n+1, which one of these will become lack of one chromosome, which is n-1. So of course, this chromosome mutation in the cell will have effects, right? So we discuss about the effect in aneuploidy in chromosomal number. Okay, so the first is in aneuploidy we have two, autosomal chromosome and sex chromosome. Okay, so in autosomal chromosome there are two effects, which is one, trisomy 21. So trisomy 21, as all of us know, is actually the Down syndrome. Okay, trisomy 21 is Down syndrome. Okay, trisomy 21 is addition in the chromosome 21. Okay, as you can see here, okay, in this circle, this is the chromosome 21. This is a human karyotype. Okay, for normal human karyotype, the 21 chromosome should be 2 chromosome. But in the trisomy 21, it have 3 chromosome. So there is an addition of a chromosome in the autosomal chromosome. Okay, and for this is very common in our society, the Down syndrome. Okay, so some symptoms that they will have is first mental retardation, and then you can see broad, flat face and slanting eyes, and also short stature. As you can see, all the Down syndrome kids usually have some similar face, just some similar face, because they all have that symptoms of Down syndrome. Okay, for monosomy 21, as you can see at the chromosome 21 is only have one chromosome. Okay, this is because the monosomy 21 it have lack in chromosome 21. So they will only have 45 chromosome. The normal human chromosome is 46 chromosome, but for trisomy 21 is 47 chromosome, while monosomy 21 is 45. So if you want to to ease your how you to memorize this trisomy or monosomy. So monosomy, mono is 1. So at 21, the chromosome 21, you have only one chromosome. So for trisomy, tri is 3. So at the chromosome 21, it has 3 chromosome. Okay. Okay, that's it for aneuploidy effect in autosomal chromosome. Okay, next we'll move to aneuploidy effect in sex chromosome. Okay, so there are two effects of aneuploidy in sex chromosome. First, we will see the Klinefelter syndrome. Okay, Klinefelter syndrome is actually occur because of non-disjunction during meiosis 1 in male. Okay, I highlight in male. Okay, so as you can see in this diagram, at first it have XY chromosome. Okay, so XY chromosome, but the non-disjunction occur here. Okay, so at this part it have XY chromosome, but at this part it have no chromosome because the both X and Y fail to segregate during meiosis 1. Okay, and then the gamete produced is XY sperm, which is for a normal person, actually the sperm will carry either X or Y. Okay, because the secondary oocyte, which ovum, will carry an X chromosome. So the sperm will carry either X or Y chromosome. So if XX, it will become a female. So if XY, it will become a male. Okay. So for this, when the fertilization occur between the XY sperm and X secondary oocyte. So this X secondary oocyte is a normal one, normal gamete. But for the normal ovum, for the sperm, it is abnormal because non-disjunction occur during meiosis 1. So it carries XY chromosome, and then if fertilization occur, it will produce a zygote of XXY. So it have an addition of X chromosome here. Okay. It actually have to become XY if male, but for this condition, it become XXY. Okay. Okay, that is for Klinefelter syndrome. Okay, let me tell you some symptom of the Klinefelter syndrome. Okay, so usually people with Klinefelter syndrome, they have poor beard growth and they have breast development. Okay, the guy, and then it will sometimes, mostly they will become sterile male because they fail to produce testosterone. Okay. Okay, that's all for Klinefelter syndrome. Now we'll move to Turner syndrome. Okay, so Turner syndrome is non-disjunction during meiosis 1, the same, but it is in female. So it is in production of secondary oocyte. Okay, so the non-disjunction occur in production of secondary oocyte. Okay, for Turner syndrome, it's actually caused by partial or complete loss of the second sex chromosome, which is X. So the one that we are focusing right now is the complete loss. Okay, so I just take one example in this diagram. Okay, first the XX chromosome, then non-disjunction occur. This part will have no chromosome at all, but this part will have two will have XX chromosome. Because the XX chromosome non-disjunction occur, it failed to segregate during that. Okay, and then the at this part, will produce gamete of XX oocyte, but at this part will have no chromosome because it failed to segregate at the first, so it will just go to this to the left part, not to the right part. Okay, so what happened when the right part with the secondary oocyte with no chromosome fertilized with a sperm? Okay, that will become here. So when the secondary oocyte with no chromosome, it fertilized with the sperm, okay, which have the normal X chromosome, okay, XX. The normal female one is XX, but this one it becomes X0, okay, because it have only the chromosome from the sperm, but no chromosome from the secondary oocyte. Okay, so the symptoms that usually people with Turner syndrome will have is first ovaries or breasts that do not develop, okay, and heart abnormalities, and the same as Klinefelter syndrome as I tell earlier is they will be sterile, okay, they will be sterile. Okay, and the last one that we're gonna discuss is about euploidy or polyploidy. So we usually say is polyploidy. So polyploidy is condition of an organism have more than two sets of chromosomes. So a normal human will have two N, but this but people with polyploidy, not people. I mean, the polyploidy condition will produce the more than 2N organism. Okay. So actually this polyploidy is common in plants but uncommon in animals. It always occur in plants. Okay. So for auto polyploidy and allo polyploidy are the types for polyploidy. Okay, auto polyploidy is actually a condition that derive from same species. Okay, but for allo polyploidy, it from the different species. So, and the difference is auto is for same species, and allo is for different species. Okay, so this is an example of auto polyploidy. So for AA and AA, so normal meiosis occur and it will produce A and E. It will have normal diploid. But this is auto polyploidy usually caused by a mutation when colchicine. Colchicine is a mutagen that will prevent the spindle fiber formation. So when spindle fiber did not form, so we can't, you know, the homologous chromosome cannot segregate to the opposite poles. It will not move to the opposite poles because the spindle fiber is not there. Okay, so the AA, the mutation of colchicine, we add the colchicine, we have mutation occur here, and it will become AAAA, which is fertile tetraploid. Okay, so now this is another part which is Aloe Polyploidy. Okay, so remember Aloe Polyploidy is from different species. Okay, as you can see here, this is a species AA, while this is a species BB. Okay, so after we undergo meiosis, a normal meiosis, it will produce A and it will produce B, and then it will the fertilization between A and B gamete will occur, and then it will produce a gamete of AB. So gamete of AB is a sterile hybrid. Okay, this is the gamete AB is a sterile hybrid because A is not homologous to B. Okay, so it cannot undergo meiosis and it cannot produce gamete. Okay, so we will so the sterile hybrid will undergo replication, okay, a doubling process. Replication is a doubling process. It will produce a fertile hybrid, AABB. So if so the replication actually occur to produce diploid AABB. Second is to ensure all the chromosomes can pair up into bivalents and produce gametes. So after we undergo replication, the fertile hybrid, which is AABB, can undergo meiosis and it will produce a gamete of AB. Okay. So that is all for my presentation. So that is for chapter 7 mutation. So I hope all of you can understand what I've been explaining from the beginning. If you have any inquiries, any question, you can ask in the comment section and we will answer it at the end of the session. Okay, thank you.
Okay, thank you, Amira. I think that was a fabulous and really amazing presentation because personally, I'm not a biology student because I'm from module 2, but still I can understand most of the points that you were trying to share just now. It's really fabulous, I've got to say. And I think I forgot to mention something to our audience just now, to our matriculation students just now, which was, I think we're going to have a Q&A session at the end of our program today at around 9:45. So if you have any question, so just leave it at the comment part and we're going to sort them and we're going to include them and we're going to put them right here at the screen so everyone can know the questions that you ask. And of course, our 4 of our leaders will work together to answer your questions and to make sure the message that we try to deliver across to you is correct. Okay. So after Amira, great job, Amira. So we're going to continue with Seema, our assistant for today. Seema, if you're ready, you can start, Seema. Oh, I think it seems as though there's some connection problem with Seema currently, but never mind. We're gonna continue with all the questions that was being asked by our friends just now. Okay, later only we continue with Seema again. Okay, so there were a few questions being asked to Amira just now when she was presenting, and so for the first question, so I'm gonna have Amira, Nicholas, and Tang together to answer those questions. And so we have a friend called Mohammad Yusuf ID Zulkifli to ask such a question. So the name of effect for monosomy 21 is what? Not an up syndrome or something?
Well, actually, I think we only address it as monosomy 21 or chromosome 21 monosomy. I don't think we call it up syndrome.
Yes.
Yeah, the reason Down syndrome is called Down syndrome is because it's named after the scientist. But for monosomy 21, there's no specific name.
Yeah.
Oh, so I got your point. So hopefully Muhammad Yusuf question is being answered. Okay, so we're gonna have something from Easy Ijan. Spinal fiber not yet appear. Is it similar to non-disjunction?
Actually, yeah. Go on.
Okay, because in non-disjunction problem, the condition is because the you have many causes of non-disjunction. So spindle fiber not appear can be one of the causes. Okay, so the non-disjunction is a condition where the homologous chromosome fail to segregate.
Okay, so I think Amira, Pang, and Nicholas has already answered. And there's a last question being added currently to the stream, which is from Nick Mukri.
Okay, Cry-Duchet syndrome. Okay,
So may I know a bit?
syndrome.
Yeah.
Okay, so Cry-Duchet syndrome is actually in the part of Cry-Duchet syndrome is actually in the part of wait a minute. Okay, in the deletion in the chromosome in the gene mutation. Okay, Cry-Duchet syndrome is a syndrome will in the deletion the base deletion problem. in gene mutation. Okay, so Craig-Duchet syndrome, one of the symptoms that I remember is the person with Craig-Duchet syndrome will have the symptom of crying like a cat, like a meowing cat.
Oh yeah, I think I remember something like we learned this in our Form 5 syllabus, I suppose, because I still have some memory about it, okay? And we have another question from Triple O. What is the name of the process to make sterile hybrid able to be produced through meiosis?
It's called chromosome doubling because before the chromosome doubling, you don't have homologous chromosome, so you cannot go through meiosis to produce the gamete. But after chromosome doubling, you double the amount of chromosome because before this, the chromosome are from two different parents, so they are not exactly homologous chromosome. But once you double already, the different chromosome can undergo meiosis, then you can produce gamete. Then only it's sterile.
Okay, so I suppose the question is being answered just now. And there's another question as well from Yingje Ku. So the replication that causes polyploidy actually occurs when? Is it during meiosis?
No, actually the replication will occur after meiosis because after the meiosis it will produce a sterile hybrid, right? Okay, so the replication will occur to transform the sterile hybrid into fertile hybrid. So it just the replication is only just a process of chromosome doubling. That's it. It's not in the meiosis.
Oh, okay. So I wish Yingje's question is being answered well by Amira. And Nick, you commented Pang is handsome. Of course, I know Pang is handsome as always. Okay, Alpha also commented Pang Senpai UWU. What does that signify? I'm not sure. And there's another question from Nana. Craig-Duchet syndrome is the addition of chromosome 5, right?
Yes, Craig-Duchet syndrome is the deletion of chromosome 5, right? Yes, in the human chromosome.
Okay, so there are plenty of questions down there at the comment section, but most of them are complimenting Pang for being handsome. That's not fair for Nick and me. That's not fair. So another question from Easy, who asked a question just now as well. Is chromosome doubling a type of mutation? Is it continuous?
No, I don't think chromosome doubling not is actually not in the type of mutation because as we learn the classes of mutation, there is no chromosome doubling. So actually, chromosome doubling is just a process to recover what the mutation have caused in the fertile hybrid.
Okay, things are getting complicated for me. I mean, like for me as a person that never studied immatriculation. Okay, I think it's the last question from Saifullah Aziz. So does it mean that every gamete in Aloe Polyploidy that undergoes replication is a fertile gamete?
Yes, because we want to transform the sterile hybrid into a fertile, so it become a fertile.
Okay. Well, there are some more questions. Okay. What does Craig-Duchet sounds like? Is it like meow meow? I suppose it's like meow meow, even though I'm not a biology student.
Yeah, maybe.
Okay. Oh, another comment from a gamer, I think. Hello, Mr. Pang. He used to teach me. He was a great teacher. He made me interested in biology last time. Take note, boys. Oh, he's very informative about the chapter reproduction. Oh,
Yes, actually, I'm going to present on that chapter later.
oh, so there's a dark humor and there's an Easter egg right down there. Okay. Okay. So last question: how to overcome sterility in hybrid? Oh, that means there are marks allocated for this question. So basically, it's a question from the books or whatever. A for
Okay, so since the hybrid is sterile, basically it's saying that one part of the chromosome, let's say chromosome A, is not homologous to B. So that's why it cannot form homologous chromosomes pairs during meiosis. So that's why it has to undergo chromosomal doubling in order to double the number of mutations. Double the number of chromosomes, so it is not an odd number. That way, it can form homologous chromosomes, can form bivalents, and then meiosis can be carried out.
Okay, another fabulous performance from Nicholas as expected. Okay, so I think that's for now. Okay, so Amira, I'm going to move you down as well as Nicholas and Pang. So that does not mean that our session has ended. We're going to continue with Seema, who quit just now because due to some connection problem with her Wi-Fi. So I'll add her back to the stream, and after this, I'm going to allow Pang and Nicholas to present themselves as well. And at the last 15 minutes of our session for today, only we will go through with the Q&A questions for once again. So Seema, take your time, and yeah, the time is.
Okay. So can you see the slide, right? Okay, so today I'll be presenting about recombinant DNA technology, which is chapter 8. Okay, so recombinant DNA technology is a technology that produces DNA molecule with segments of different sources or by combining gene from different organisms. That means as long as there's combination of gene, it's called recombinant DNA technology already. Okay, so now we'll look at the 5 tools. Like in exam, when they ask the tools mean, you have to write this 5 things. It's more like the element, like you need all these 5 things for recombinant DNA technology to work. Okay, first of all is target DNA. Target DNA is like the DNA segment. Like in this slide, I'll use all insulin production examples. Okay, everything, all the examples are from insulin production. So let's say now I want to do insulin. Okay, so your target DNA will be the DNA segment which produces insulin. Okay, that will be your target DNA. And then this target DNA it needs to clone itself, right? You cannot just let one DNA and then ask them to do a lot of insulin, cannot, right? So you need to clone it. So that's where DNA cloning vector comes in. Okay, for insulin, the DNA cloning vector is plasmid. Okay, so then this DNA cloning vector, it needs to be transformed into a host cell. Because you just let the DNA cloning vector be there, then where will it find amino acid all? It needs one cell, right? Then only easier, so it will transform into a host cell. Okay, then this restriction enzyme is like just now I say it's just a segment of DNA that you need, right? So we have the DNA molecule, then we need to cut it. We need to cut out one DNA segment. So that's where restriction enzyme comes in. Restriction enzyme is like the scissors. And then DNA cloning vector also needs the help of restriction enzyme because this target DNA will be inserted into the cloning vector, kan? Then it needs some space to be inserted, lah. Like the visual will help later. And modifying enzyme, I will elaborate on the other slides. Okay, first of all, cloning vector. Cloning vector is something that can carry foreign DNA in host cell and replicate there. Like what I said just now, it replicates. Okay, so you need to fulfill 3 requirements to become like cloning vector. First of it is you have to be able to accept foreign DNA. So plasmid, the cloning vector for insulin production, has this MCS. So during exam, you cannot write MCS. You have to write multiple cloning site. Okay, so this we have the MCS, and then in this MCS, the target gene will be inserted. Okay, and then second requirement is you must be able to replicate independently in the host cell. That means the host cell mustn't influence whether the gene of interest can be replicated or not. You see, it has this origin of replication, can't? So you don't need the host cell to come and say, hey, go and replicate. No, it can replicate by itself. And then the third requirement is it must possess selectable genetic marker. This one I'll explain further in other slides. Moving on, we have restriction enzyme. Restriction enzyme is an enzyme that recognizes specific base sequence, then cut the DNA at specific site to produce DNA fragment. Okay, So I'll be focusing on two enzymes only. There is EcoR1 enzyme. One of it is EcoR1 enzyme. So this EcoR1 enzyme produces sticky end fragments. Okay. See, this EcoR1 actually cuts the DNA molecule at different positions. So we have several nucleotides overhanging here. So once there is complementary base pair, it will just stick to it. So hence the word sticky end lah. And then so these will be like the target gene. Like what I said just now, this cloning vector also need to be cut. Then only will have space for it to come inside. So both of it needs to be cut using restriction enzyme. Okay, once the target gene is inserted into the cloning vector, it will be called recombinant DNA molecule. So since this is an enzyme, so enzyme A will be specific. So for EcoR1, it recognizes the specific base sequence GAA-TTC. Okay, this sequence, we call it palindromic sequence. So what is palindromic? Palindromic means like the base pair is read the same on complementary stand in opposite direction. Let me see here 5' to 3' GAA TTC. Again here 5' to 3' GAA TTC. See, I'm telling the same thing, right? So that's palindromic sequence. So if you still don't understand, you take one paper, then you write down that EYE. Then you spell it backwards. Then you read both of it, you'll find that you're reading the same thing. That's palindromic. You still don't understand, you can try with now I want or no lemon, no melon. Okay. Moving on, another one more enzyme is the SMA1 enzyme. So the SMA1 enzyme detects this base sequence CCCGG, and the end type is blunt end because it cut at the same position. So there's no nucleotide overhanging each other or what. Okay. And moving on to the host cell. Host cell is the organism that has been used to receive a recombinant DNA. Okay. So the characteristic of host cell is first it must be able to receive a DNA during transformation process. Transformation is like you take the plasmid, you put inside the host cell, that is transformation. Okay, so it must be able to receive. The host cell must be able to receive. And then it must be able to maintain the structure of DNA recombinant from one generation to another generation. So let's say the first generation, the host cell will produce insulin. Second generation, the host cell go and produce keratin. You don't want that. You want every generation all produce insulin. That's what we mean by that. And then another one more is to amplify gene product from DNA recombinant. I mean, it must be able to reproduce. Cannot just one cell go and do all the insulin and give everybody, cannot. Like it must be able to reproduce, and then the gene product, which is insulin, must have a lot though. And then antibiotic resistance, I'll explain in the other slides. Okay, so moving on, we have modifying enzyme. So there's two type of modifying enzyme. One is DNA ligase. DNA ligase catalyzes the formation of phosphodiester bond between sugar phosphate backbone. This is just like just now I say the target DNA will be inserted into the cloning vector kind. So this DNA ligase is just to like seal it. That's all, just to seal it. So this one, if you want to know more about this one is actually in chapter 1. Okay, you can look through chapter 1. And then we have TAQ DNA polymerase. This is used in polymerase chain reaction. So this polymerase chain reaction is used to duplicate the gene of interest. Okay, and then this TAQ DNA polymerase is heat resistant enzyme. So PCR is done in high temperature and this thing is heat resistant. That's all. Okay, moving on, these are the steps for recombinant DNA technology. So first of all, we have isolation of gene, cleave/cut, insertion, transformation and amplification, and then screening. Okay, so actually this one they will ask a lot in exam, like in the question papers. They will ask like give a picture and then they'll ask like which step is it. Okay, so isolation is just you isolate the gene of interest and plasmid. I mean the plasmid will actually be in a bacterium, like for insulin production lah. The plasmid is U18 and the bacterium will be E. coli. Okay, so you have to isolate the plasmid and then you have to isolate the gene of interest, the DNA segment for insulin. Okay, moving on, we have cleave or cut. Okay, so the target gene and the vector must cut with the same restriction enzyme. Okay, so let's say if your vector you use cloning vector you use EcoR1, your target gene when you cutting it time also you must use EcoR1. Then only you can produce complementary sticky or blunt end. I just want to highlight something. For insulin production, when you insert the target gene, it will be inserted inside the lag Z gene. Okay, you all just remember this. will be inserted inside the leg Z gene. Okay, next we have insertion. Insertion is the target gene will be inserted into the plasmid. That's all. This target gene inserted into plasmid. And then ligation also occurs. Like sees the strand, just now I said during modifying enzyme. So same thing, DNA ligase catalyzes the joining process by forming phosphodiester bond. Okay. Some plasmid will be unable to join with target DNA and they are called non-recombinant plasmid. See, this one already joined, right? So it's called recombinant plasmid. Then those who didn't join, it's called non-recombinant plasmid. That's all. Moving on to the transformation process. Transformation is just the plasmid introduced into the host cell. Okay, so there's actually three consequences. One is the host cell will receive recombinant plasmid. Another one more is the host cell will receive non-recombinant plasmid. I mean, this one don't have the target gene. And then another one more, no plasmid. The host cell won't receive anything at all. Okay. And then we have amplification. Amplification is the process in which the bacteria is allowed to reproduce. Okay. So this is just like, this now I already transform it here, right? So everything will reproduce. No matter you got recombinant plasmid, you know plasmid, everything all reproduce. And then it will be played out on the bacteria on solid nutrient medium containing ampicillin and sugar agar. Remember, ampicillin and sugar agar. Okay, so next we move to the screening process. So the screening process for insulin production is blue-white screening method. This is for insulin production only. Okay, like others will have different methods. Okay, so before we go deep into it, let's look at the basic first. So the plasmid, this POC18, has this AMPR gene. Okay, so what's the importance of this AMPR gene? This bacteria without this gene cannot survive when put in a medium with ampicillin. So remember just now I said in that medium got ampicillin, right? And then during transformation process, one of the consequences is no plasmid will enter the host cell, right? So the bacteria with no plasmid, once you put inside the nutrient medium already, will die because they don't have AMPR gene. Okay, so this plasmid also contains LAC-Z gene, the place where the target gene is inserted. This LAC-Z gene encodes for beta-galactosidase. This one is actually from chapter 6, the lac operon, that stuff. Okay, so LAC-Z gene encodes for beta-galactosidase. So this beta-galactosidase hydrolyzes a mimic of lactose. I mean, it's like lactose but not lactose. Okay, mimic of lactose, also called agar lah. Remember I said just now in the medium got agar. Okay. So now we already the bacteria with no plasmid, the one already died. Now we are looking at recombinant plasmid. Okay. Recombinant plasmid has non-functional LAC-Z gene. Why I say like this? Because the target gene will be inserted in the LAC-Z gene. So once they're inserted, they're disrupted. So beta-galactosidase not produced, unable to hydrolyze X-cal sugar, will form white colony. For non-recombinant plasmid has functional LAC-Z gene, means intact, nothing insert kan, so it's intact. So beta-galactosidase is produced and able to hydrolyze X-cal sugar and form blue product. Okay, so blue product is non-recombinant plasmid. White colony is recombinant plasmid. Okay. Okay, move on. This is the summary. Like during exam, like they will they'll tell you like they will remove all these red words and then they give you a box and then tell okay wait which step is this, which step is this, which step is this. During exam they ask things like that. Okay, so now we going into the details of insulin production. So the host cell is E. coli. And the gene of interest is from human pancreatic cell in the form of cDNA. Okay, I'll explain further about this. Okay, since it's taken from human pancreatic cell, we don't have many compatibility issues. Okay, because it's from human pancreatic cell. Human, no compatibility issues. Okay, and then bacteria with recombinant plasmid is not entirely right. One with white colonies, they all will be placed in the fermentation tank to multiply. And then the gene will undergo transcription and translation. This is from chapter 6. The gene will undergo these processes and then it will produce insulin. Then you write up insulin, then you can give everybody. Okay, so now I'll focus on cDNA. Okay, so first of all, from the human pancreatic cell, we have the mRNA. Okay, then this reverse transcriptase, which is an enzyme, catalyzes the synthesis of a single cDNA strand. I mean, it helps in synthesizing the single cDNA strand. And then we have an mRNA degrading enzyme to degrade this mRNA. And then DNA polymerase will come, synthesize second DNA strand complementary to single-stranded cDNA. So kita dapat cDNA dah. Okay, so a little bit about cDNA, a summary about cDNA. cDNA is synthesized from mRNA, and it is catalyzed by reverse transcriptase and DNA polymerase. I'll just explain this just now. Okay, so why you don't want to straight away take from mRNA? It's actually because mRNA represents expression of gene with introns. The cDNA represents expression of gene without introns. So what is introns? Introns is like the non-coding segments of DNA. Like these introns won't produce any protein. Okay, like it's useless. It's like useless. For human, we are what? We are eukaryotic cells, right? So we have RNA processing, a function also called RNA splicing. This function will remove all these introns. So when we are doing the protein, everything, the protein amino acid sequence, all not disrupted, all bagus. But then for bacteria cell, is prokaryotic. It don't have this function. So when we are inserting this gene, we must make sure that everything like don't have intron. Okay. So that is all from me.
Okay, I think that was also a really fabulous presentation from Seema. As things are getting complicated right away, I've got to say. As the subtopic proceeds further and further, I'm getting lost. Sorry, because I'm not a biology student anyway. But I'm sure all of you inside the chat, for those that are looking at this stream, they understand your presentation really well. Okay. So I'm going to proceed with Nicholas first. Only then, after Nicholas and after Pang, only then we will conclude all of your questions inside the comments. But whoa, there's a statement. Seema is so pretty. Of course, Seema is pretty. Everyone in KMS is very pretty and very handsome, except me. Okay. Okay. So now we'll continue with Nicholas. Yeah. So Nicholas, take your time and show me what you got.
All right. Can you all see the slide screen? Right. Right. So my name is Nicholas, and today I'll be presenting Chapter 9, Reproduction and Development. I'll be starting with the first subtopic, which is reproduction in plants. So before we start with the formation of male gametes, I would like to talk about basically the structure of the male reproductive organs in flowering plants, also known as angiosperms. So the male organ of a flowering plant consists of one or more stamens, or which you can see in the diagram over here over here. Yeah. It is made out of one or more stamens, and each stamen is made out of an anther, which is the top part, and the filament, which is basically like the stalk. So each anther has a sac that contains a pollen sac. So an anther and a filament together will make up a stamen. So pollen grains are produced in the anther, and each anther consists of 4 pollen sacs, also known as microsporangia, which are the dark yellow circles over here. So each pollen sac has many diploid cells, known as microsporocytes, also known as microspore mother cells. Each microsporocyte will undergo meiosis in order to form 4 haploid microspores. Each microspore divides once by mitosis in order to form a pollen grain. And a pollen grain consists of a generative cell and also a tube cell. So based on the diagram here, you can see that these are diagrams taken with an electron microscope when they zoom it like around 400 times into a plant. And this is how the pollen grain looks like. And these tiny hairs are what is basically the structure that they use to latch onto the style of a female flowering plant. So the pollen grain has a thick and tough resistant outer wall known as the axene. All right. So when a pollen grain when a pollen grain lands onto the stigma of a carpel, it will turn into a mature male gametophyte. Then only will the generative cell start to undergo mitosis in order to divide into 2 sperms or male gametes, and a pollen tube will be formed. But we'll be talking more in depth about this in double fertilization. All right, next we'll be talking about the development of the embryo sac and the formation of female gametes. So the formation of a female gamete starts with the development of the ovule within the ovary. So an ovule consists of three parts, which is the integument, the nucellus, and also the megagametophyte, also known as the embryo sac. So based on this diagram here, you can see that the inner part is known as the integument, while the outer part of the integument is known as the outer integument. The hole in the bottom here for the sperms to enter during fertilization is known as the micropyle. While the embryo sac is the part in the middle here. The ovum is the cell surrounded by two synergid cells, while there are three antipodal nuclei on top of the embryo sac. The nucellus is the space inside the embryo sac. An ovule is also attached to an ovarian wall by a short stalk known as the funicle, or plural known as funiculus. So during development, the nucellus acts as the food reserve for the ovum. The nucellus is formed from the placenta of the ovary. The base of the nucellus will develop into two layers of integuments, but it will leave an opening called the micropyle at an end. The integuments basically function as a protective layer as they later develop into the seed coat, while the micropyle is a passage for the entry of the pollen tube of fertilization so that the sperm can fertilize the ovum and fertilization can take place. The chalaza is the place where the nucellus fuses with the integument. The embryo sac formation occurs in a tissue known as the megasporangium, which is found inside each of the ovules. So there's one cell in the megasporangium, also known as the megasporocyte, which is diploid, undergoes meiosis in order to form 4 haploid megaspores. 3 of the megaspores, which are smaller in size, will degenerate and will be absorbed into the nucellus, while the one remaining megaspore will divide 3 times by mitosis without undergoing cytokinesis. So it will basically form one big cell containing 8 nuclei. This will produce an embryo sac. So 3 cells will migrate to the chalazal end to form antipodal cells, while another 2 nuclei will stay at the middle to form the polar nuclei, which later develop into the triploid endosperm after fertilization. The 3 other nuclei migrate to the micropylar end to form 1 egg cell in the middle and 2 synergid cells next to it. So next we'll be talking about pollen, germination, and also double fertilization. So when a pollen grain lands onto a stigma, the glucose and moisture on top of the stigma will stimulate the pollen grain to germinate. So the germinating pollen grain will form a pollen tube that extends down the style into the ovary. As the pollen tube grows, the generative nuclei inside will undergo mitosis, producing 2 sperm nuclei or male gametes. Now the pollen grain is known as a mature male gametophyte. So the pollen tubes will carry the 2 male gametes as it moves. At the tip of the pollen tube, when it enters the ovary, it will discharge 2 of the sperms into the embryo sac. One of the sperm will fertilize with the ovum to form the diploid zygote, which later develops into an embryo, while the other sperm will combine with the 2 polar nuclei in the middle to form a triploid endosperm nucleus in the center of the large central cell of the embryo sac. The large central cell will develop into the endosperm. Endosperm is the food storing tissue of the seed, basically for the development of an embryo. The union of 2 sperms with the different nuclei in the embryo sac is known as double fertilization because fertilization occurs twice, once at the ovum and another time at the polar nuclei in the middle. So after double fertilization, the ovule will develop into the seed while the ovary develops into a fleshy fruit. The wall of the ovary will develop into the pericarp of the fruit. The integuments will develop into the seed coat, also known as the testa, while the micropyle remains as a tiny hole in the testa. So usually in exams, they don't really ask the definition, but just for your knowledge, double fertilization is defined as the complex fertilization mechanism that involves 2 sperm cells, One fertilizes the egg to form the zygote, while the other fuses with two polar nuclei to form the endosperm. And the suspensor is found in plant zygotes in endosperm, and it connects the endosperm to the embryo and basically provides a route for nutrition from the mother plant to the embryo. While the proembryo is the cluster of cells in the ovule of a fertilized flowering plant that has not yet formed into an embryo. The life cycle of a flowering plant is the next part. So the transfer of pollen grains onto the stigma is known as pollination. If the pollen grains are transferred to the stigma of the same flower, it is known as self-pollination. Likewise, if it is transferred to the stigma of another flower but of the same species, it will be known as cross-pollination. So cross-pollination will cause a genetic variation in the species and will cause the offspring to be slightly different from the parent plants. This method of pollination does not require any investment from the plant to provide nectar and pollen as food for the pollinators. This type of pollination have been studied ever since the time of Gregor Mendel. I'm pretty sure you've heard of the name Mendel. All right, the next part is the formation of the seed. So as the sporophyte embryo develops from the zygote, the seed will start to store more proteins, oil, starch, and other minerals, which is why seeds are basically storage for nutrients for the seed to develop when it is planted. When mature, a seed will consist of a dormant embryo which is surrounded by food and protective layers, which used to be the integuments of the cell. So to summarize everything, a double fertilization involves two sperm cells. One fertilizes the egg cell to form a diploid zygote, while the other fuses with two polar nuclei to form a triploid endosperm. After fertilization, the fertilized ovule will form the seed, while the tissues of the ovary become the fruit. Right, next we'll be moving on to the next topic, which is the reproductive system of humans. The female reproductive system can consist of a main part known as the ovaries. The ovaries are oval in shape, about 4 centimeters long, and they lie on either side of the uterus of the wall, in a region known as the ovarian fossa. The main function of the ovaries are to produce oocytes, which are the female gametes, for fertilization, and also to produce female sex hormones, estrogen and progesterone, in response to the pituitary hormones LH and FSH. The structure of the ovary is covered by germinal epithelium cells, and the cortex of the ovary has many stages of follicles, known as the primary follicle, which is the smaller follicle, which later develops into a secondary follicle, which is the larger version, and the Graafian follicle when it is ready for ovulation. Each follicle only contains one egg cell, and the follicle will nourish and protect the egg cell during development. When the follicle is mature, it will release the secondary oocyte for each menstrual cycle. So usually only one secondary oocyte is released each month. So the structures involved are the germinal epithelium, which is the surface of the ovary, the primary follicle, which contains the primary oocyte surrounded by the zona pellucida, which later develops into the secondary follicle, which develops antrium and has an outer layer known as the theca. The secondary follicle will develop into a larger Graafian follicle, which contains a secondary oocyte and the first polar body, which later degenerates. There's a lot of granulosa cells surrounding the oocyte. Zona pellucida is known as the corona radiata. Next is also the corpus luteum, which is formed from the follicle after ovulation occurs. So the structure of a secondary oocyte is the structure of a secondary oocyte can be seen from the diagram below. It has a haploid nucleus in the cell and contains numerous vesicles known as the cornical granules. The zona pellucida is surrounded by the corona radiata, which is basically made up of many layers of follicle cells. All right, so these are the names of the hormones that we'll be using in the female reproductive cycle. GnRH, the gonadotrophin releasing hormone, LH, luteinizing hormone, and also FSH, follicle stimulating hormone, estrogen, and also progesterone. So LH and FSH are secreted by the pituitary gland, while the estrogen and progesterone is produced in the female reproductive system, while GnRH is produced by the hypothalamus. So the roles of female hormone in the reproductive cycle is as follows. The follicle stimulating hormone, FSH, is stimulated by the anterior pituitary gland, which will promote the growth of follicles into secondary follicles and later into Graafian follicles. Estrogen is secreted by the follicles I mentioned before. The estrogen secreted by the growing follicles during the follicular phase, and its function is to repair and thicken the endometrium wall. Next is GnRH, which is the gonadotrophin-releasing hormone, is secreted by the hypothalamus. Its function is to stimulate the anterior pituitary gland to secrete more FSH and LH, depending on the levels. LH is the luteinizing hormone, and it triggers ovulation to occur. It also results in the formation of corpus luteum, which later produces estrogen and progesterone. However, corpus luteum produces a lot more progesterone compared to estrogen. Progesterone is also used to stimulate the thickening of the endometrium wall to get ready for implantation of an embryo if fertilization occurs. Progesterone also inhibits LH and FSH during the luteal phase. Right, I'll be moving onwards to the stages in oogenesis, which is the process of production of male-female gametes from the oogonia into ovum in the ovaries. So oogenesis begins soon after fertilization. So the primordial germ cells in the ovary begin to undergo mitosis and differentiate into oogonia. Most of the oogonia die, and the remaining that survive will divide by mitosis in order to form primary oocytes. Primary oocytes, which are still diploid at this point, will go through meiosis 1, but stops at prophase 1 until puberty occurs. So the steps before here, from step 1 over to step 3, they all occur during the the infant phase of a female. The primary oocyte will start to develop into a secondary oocyte during puberty once they complete the process of meiosis 1. A larger cell, known as the secondary oocyte, and a tiny one, known as the first polar body, which are both haploid, are produced. The secondary oocyte will begin meiosis 2 and stops at metaphase. This is when ovulation will occur. If fertilization occurs, meiosis 2 will be completed, and when the sperm fuses with the secondary oocyte, it will produce an ovum and a second polar body. The next subtopic is the male reproductive system. The main functions of the male reproductive system is to produce, maintain, and transport sperm and their protective fluids known as semen. It will discharge sperm into the female reproductive tract, and it will produce and secrete male sex hormones such as testosterone. There's two types of male reproductive system, which is the external one, which you can see in the diagram here, this long thing here, and the two round objects around it, and also an internal reproductive structure, which consists of the prostate glands, the seminal vesicles, and vas deferens, and everything else in the diagram here. Right, the external reproductive structure is made up of the testicles. So most men have 2 testes. So yeah, if you're considered normal, if you have 2, and it is responsible for making testosterone, the primary male sex hormone, and also produces sperm. Within the testes are coiled masses of tubes known as the seminiferous tubules. The epididymis is basically this tube over here. It's the beginning part of the tube. It carries and stores sperm cells that are produced in the testes, and then it is the place where the sperms are stored until they become mature and motile. Next is the scrotum, which is the pouch-like sac of skin, the wrinkled part of under the penis. It contains the testicles and has many nerves and blood vessels. Therefore, when you get hit in the nuts, it really hurts because there is a lot of pain receptors over there. It has a protective function and acts as a climate control system for the testes, which shrinks when it's cold and it will expand when it is hot. Next is the internal reproductive structure. The first part is the urethra, which carries the urine from the bladder to outside of the body. in males, during ejaculation, it also expels the semen when the man orgasms. Next is the prostate gland, which is this purple color walnut-sized structure in the middle under the bladder. It produces fluid that nourishes and transports the sperm. The vas deferens is the long muscular tube that travels from the epididymis into the pelvic cavity. It's located behind the bladder. It carries the sperm to the urethra. And lastly is the seminal vesicles, which are sac-like pouches that are connected to the vas deferens. They produce fructose, which provides the sperm with energy and helps them to move towards the ovum when for fertilization. All right, next is the structure of spermatozoa. Spermatozoa consists of three main parts, which is the head, the midpiece, and also the tail. The head is a smooth or an oval shape which contains the nucleus. It's haploid, so it only contains 23 chromosomes. It is made up of several parts that include the acrosome, which secretes hydrolytic enzymes. Next is the middle piece. The middle piece is basically the neck of the sperm, and it contains many packed mitochondria so that it can produce energy it needs in order to swim towards the ovum. And lastly is tail, which is basically the part needed for motility of the sperm. It contains arrangement of 9 microtubules. All right, for the hormones which are involved in spermatogenesis, the first hormone is GnRH, LH, and FSH, which are all similar to the females. So GnRH has the same function, which is to stimulate the anterior pituitary gland to secrete LH and FSH. However, the function of LH differs in males. Instead of stimulating progesterone to be produced, LH will stimulate the Leydig cells to secrete testosterone, while follicle-stimulating hormone FSH will stimulate the production of sperm, which it also stimulates the Sertoli cells to secrete inhibin when the level of testosterone is too high. Next is inhibin. Inhibin inhibits the anterior pituitary gland to reduce the secretion of LH and FSH. Testosterone, however, is used to stimulate spermatogenesis, and it is basically the main sex hormone which is responsible for the development of male characteristics. All right, next is the role of, first, the process of the hormones works as follows. So the hypothalamus will first secrete GnRH, and the GnRH will stimulate the anterior pituitary gland to produce hormone FSH and also LH. FSH will act on the Sertoli cells, which nourish the sperm, and the LH will stimulate the Leydig cells to produce more testosterone. Then this testosterone will basically produce sperm, and it will also control the growth of the vas deferens, testes, scrotum, and penis, and also stimulate sexual urge. Right, next is the stages of spermatogenesis, which is the process of producing mature sperm cell from spermatogonia. It's a continuous process that occurs in adult males after puberty. So unlike oogenesis is only a temporary process because it stops after menopause occurs around the age of 55. It occurs in the seminiferous tubules of the testes. Right, so as you can see from the diagram, it starts off from the primordial germ cells, which undergo differentiation in order to develop into a diploid spermatogonia. And the spermatogonia will undergo mitosis to develop into two smaller daughter cells. One of them remains as a spermatogonia, while the other develops into a primary spermatocyte, which is diploid, to prepare for meiosis. So this usually occurs around 12 to 14 years after birth when a male enters puberty. Next, the primary spermatocyte, which is diploid, will undergo meiosis in order to form two secondary spermatocytes, which are both haploid. So the process takes approximately 9 days, and the secondary spermatocytes will continue to undergo meiosis too in order to produce 4 spermatids, which are all haploid. So the process takes about 19 days. So the spermatids will obtain nourishment from nearby Sertoli cells in the seminiferous tubules for them to keep growing and develop. Eventually, the spermatids will undergo cell differentiation to become mature sperm cells, spermatozoa, where they have fully developed heads, midpiece, and tails, so they can do their function. There's a mistake here, and it's supposed to be haploid spermatozoa, not diploid. Next is the hormonal control of the male reproductive system. So there are two main negative feedback mechanisms which control the production of hormones in the males. So when GnRH is secreted to the anterior pituitary gland, it will stimulate the production of LH and also FSH. When there's LH in the male reproduction system, the Leydig cells will produce testosterone, which is needed for spermatogenesis. So testosterone will produce a negative feedback mechanism to reduce the production of LH. So this process is a repeatable cycle, because when the production of LH is decreased, the levels of testosterone also decrease. And then when testosterone decreases, it won't reduce the production of LH anymore, so more LH will be produced. So it's basically a continuous cycle in order to maintain the level of testosterone at optimum levels in the male. The second part is when the FSH stimulates the secretion of inhibin by the Sertoli cells. So the inhibin will act on the anterior pituitary gland to reduce the production of FSH. So the negative feedback mechanism will maintain the level of hormones at a normal range. So thank you, and that will be it for my part. And Tang will be continuing with the rest of the chapter from 9.3 to 9.4.
Yeah, I've got to say again, thanks Nick, is really fabulous, I've got to say, because the way you explain everything is so clear, and even also I noticed like those things that you wish that you discussed with all of us just now are the things that we learned in Form 5, of course more in a more advanced version. And I can remember a lot of things inside, like Gregor Mendel, did he propose the Mendel model? I think he proposed the Mendel model, is it?
Yeah, that was him.
Yeah,
the
okay.
Mendel first law and second law.
Yeah, that thing that drove me crazy when I was in Form 5. Okay. So I'm going to continue with Pang currently. Okay, so Pang, take your time and show us.
Let me just share my screen first. All right, I hope you guys can see this. So now I'm gonna present about my favorite chapter, reproduction. I mean the fertilization part, which is under reproduction. So first, I'm gonna be talking about fertilization. So we need to understand these two subtopics I'm gonna be talking about later, because it's really popular in essay questions, and I've observed the question patterns, and consistently at least one of them will come up. So first, we need to understand what is fertilization. So fertilization is basically when two haploid gametes that will fuse together to form one diploid zygote. So in this case, we're talking about human fertilization. So therefore, the gamete would be a sperm cell and a human egg. So as we can see, as Nicholas explained earlier, sperm cell is this thing with three parts: the head, midpiece, and tail. And human egg is the secondary oocyte. Okay. So I will explain a bit about sperm structure again, because later we will be using it. So the first part is the head, which contains nucleus, which holds the genetic information of the father, and acrosome, which contains hydrolytic enzyme. Then the middle piece contains mitochondria, which provides energy for the sperm to propagate forwards and swim towards the secondary oocyte and fuse with it. The last one is the tail, which allows the movement of the sperm. So when does fertilization occurs? It occurs when the freshly ejaculated sperm enters the vagina. It occurs the moment the sperm enters the vagina. So it will start the four phases of fertilization. The first stage is the capacitation. So when the sperm is ejaculated into the vagina, capacitation occurs. So what is capacitation? Capacitation is a necessary process that must occur for the sperm to successfully fertilize and one secondary oocyte. So basically, this process is to increase the function of the sperm. So as we all know, the function of a sperm is to fertilize the egg, but sperm cannot successfully fertilize egg without capacitation. And some of you might ask, why is that? Because capacitation is a process where it enhances the function by allowing sperm to sense the chemical signal released by the secondary oocyte. So it is able to achieve that by, if we look at this diagram here, we have the sperm, and then the head here actually you can see cholesterol and glycoprotein. So in exam, you can just write the capacitation process removes the component on the plasma membrane of the sperm. So you can imagine this. You are wearing a mask and you cannot actually smell that much things, right? So same thing with the sperm. With all these components on the sperm plasma membrane, which is the head here, it cannot exactly detect something far away, cannot detect it effectively. Therefore, capacitation allows these components to be removed, and it's like you taking down your mask and you can smell something further away. And same concept with the sperm here. The moment these components are removed, the sperm can effectively sense the chemical signals released by the secondary oocyte. Therefore, it can swim towards it immediately. The second thing that the capacitation process does is it increases the motility of the sperm. So how does it work? Is like same thing. As these components are removed, the motility of the sperm will be increased as they can swim more effectively and further. Since the sperm can sense secondary oocyte from further away from just the vagina, therefore it can instantly swim to it without wasting any energy or time. Okay. Then for one more thing is that when these components on the sperm head is removed, the sperm can fuse with the egg. So later we will talk about why does it need to fuse. But basically, with all these components blocking, the sperm is unable to fuse with the secondary oocyte. So capacitation is a very important process. Next, after the sperm is capacitated, the capacitated sperm will start to move towards the secondary oocyte. And keep in mind, at this point, we don't address the egg as ovum, but we need to address it as secondary oocyte. So the sperm will move towards the secondary oocyte, and as it approaches the secondary oocyte, and we need to look at this diagram here. This is the female egg structure. So the outer layer is a corona radiata. What it does is it just provides nutrients for the secondary oocyte. It does nothing. So the sperm can just go past this corona radiata. But the problem starts when the sperm approaches the zona pellucida, which is the jelly coat layer that protects the secondary oocyte. The sperm cannot just penetrate the zona pellucida because the zona pellucida kind of just seals off this area. So in order for the sperm to penetrate, on top of zona pellucida, there's a component that acts as the sperm receptor. So sperm can bind to it. The sperm will bind to the sperm receptor, and when it binds, right, the acrosome that I mentioned earlier, the acrosome which contains a hydrolytic enzyme, it will rupture, and when it ruptures, the hydrolytic enzyme will be released onto the surface of the zona pellucida. So when this happens, right, the hydrolytic enzyme will digest through the zona pellucida, creating a pathway for the sperm to enter. So when this sperm digests ready, it instantly triggers the next phase. The next phase is the fusion of sperm head membrane and secondary oocyte. So the layers are as like corona radiata, zona pellucida, then only the sperm can reach the plasma membrane of the secondary oocyte. So after the digestion of the zona pellucida, the sperm moves towards the plasma membrane, and this is unlike zona pellucida that don't need a receptor, so sperm can just fuse with the plasma membrane of secondary oocyte. Both the plasma membrane will fuse, and if we look at the diagram here, this purple molecule is the nucleus of the sperm. When the sperm fuses with the secondary oocyte, the purple molecules is released into the inner content of the secondary oocyte. So after it fuses, it's as if the door is open and the nucleus is allowed to enter into the secondary oocyte. And there's not much about this fusion, but we need to keep in mind that the moment the sperm fuses with the secondary oocyte, it instantly triggers two events. One of the events is, as I said earlier, you cannot say secondary oocyte, you cannot mention the thing as ovum from the start, because one of the events is now the secondary oocyte will be triggered to complete their meiosis 2. As Nicholas explained earlier, they start meiosis 2 but stop at metaphase 2. But the moment the sperm plasma membrane fuses with the secondary oocyte plasma membrane, the meiosis 2 is triggered to continue. So from the metaphase 2, it will finish the whole meiosis process, and therefore it will form an ovum. Then this ovum, right? The next thing is the release of the the fusion of the plasma membrane of these two gametes will also trigger cortical reaction, which is the last phase. This cortical reaction is not a lot, but basically it's just after the plasma membrane, you will find okay, if you look at this diagram here, this small round ball thing, these are actually cortical granules. Wait, yeah, these are actually cortical granules. So in cortical reaction, after the fusion of the plasma membrane and the plasma membrane of sperm head and the secondary oocyte, the cortical granule will instantly release this enzyme, will instantly release a type of enzyme which is sent towards the zona pellucida of the secondary oocyte. And as we learned in chapter 1, if I'm not mistaken, the process of excreting a certain liquid out from the plasma membrane of a cell is called exocytosis. So same thing here, since from the inside the cell released this enzyme and it's passed through the plasma membrane towards the zona pellucida, right? So this is called exocytosis. This enzyme will actually cause the zona pellucida to harden, and it causes the sperm receptor on the zona pellucida to prevent any more sperm from binding onto the sperm receptor. And this process occurs very fast. So one sperm fuse, and then all the chain reaction occurs, and then instantly cut off the binding of the sperm. So now it's the zona pellucida will harden and prevents any more sperm from penetrating. This is very, very important because just imagine if more than one sperm get to enter the secondary oocyte. And that also means more than one sperm nucleus can fuse with the secondary oocyte nucleus. And then from haploid, one end, you will form the secondary oocyte, one end will fuse with a few sperm nucleus. That's like, you can cause like 10N or something. You can cause the zygote to die. So in order to prevent that from happening, cortical reaction needs to occur. Then after while the cortical reaction occurs, the sperm head earlier, this one, the first sperm nucleus that is released into the secondary oocyte fuses with the nucleus of the secondary oocyte. Then it will form a zygote, and then this zygote is ready to undergo the first cell division to produce like embryo and other stuff. Then next, I will be talking about roles of hormone during pregnancy and parturition. So for those who don't know, pregnancy is basically the 9 months before you were born, and parturition is when the delivering process is occurring. So when you are giving birth, that is called parturition. Okay, we need to know that pregnancy is divided into 3 different trimesters. The first trimester is the first 3 months, so that's why people say pregnancy is typically 9 months, because it's divided into 3. There's 3 semesters, and there's 3 trimesters, and each trimester got 3 months. So the first day of the trimester is actually the day of the menstrual cycle, the first day of the menstrual cycle when you first bleed and stuff. Okay, so we'll be talking about each trimester and what occurs in those trimesters. The first trimester, you will realize that. Oh yeah, look it. Okay, so if you look at this diagram here, this is the weeks of pregnancy. So about here, like around here, is your first trimester. Then about here is your second trimester. So if we pay attention to this box, right, this area, the first trimester, the human chorionic gonadotropin is very, very high. So actually, why do we need it? So we need to understand the function of the human chorionic gonadotropin. It's actually released by the chorion, and this human chorionic gonadotropin helps maintain the corpus luteum. As Nicholas explained earlier, if no fertilization occurs, the corpus luteum will degenerate, will turn into corpus albicans and degenerate. So now that the fertilization occurs, you must maintain the corpus luteum there. How do you do that? It's with the presence of human chorionic gonadotropin. And why do we need the corpus luteum for now? Because corpus luteum will secrete estrogen and progesterone. Estrogen and progesterone can maintain the thickness and thickening of the endometrium wall. And we need the endometrium wall the thickness to maintain, because we need the implantation of the zygote. because with the implantation, then only you can grow and have a baby and stuff. So human chorionic gonadotropin is very high in this first trimester. And we will actually go into detail why is it low in second and third trimester later. So progesterone is to maintain the thickening of the endometrium wall, and estrogen is to thicken the wall. Okay. And then, okay. There's also other function for estrogen, because later on into pregnancy, the wall is thick enough already. It's not just needed to thicken it some more. Okay, at some point you will need to stop thickening and just maintain the thickening. So estrogen can actually protect the female fetus from the effect of the mother's androgen. So androgen is actually a hormone that allows male babies to develop their masculinity. So let's just say if you are a girl, you don't want yourself to have like the boys' secondary sex growth, right? As in, like, you don't want to have all those muscle, and for girls maybe puberty, they hit puberty, they have like thick thighs and stuff. And for boys, you will start growing muscle, mustache and stuff. So androgen actually causes you to have mustache, your muscle to grow, and you don't want that from female fetus in a girl. Therefore, estrogen will protect the female fetus only. So male fetus, you will not have any effect. Male fetus wants the androgen. Okay, and then now we look at this. We finished with first trimester already. First trimester, if exam asks, you can just explain the hormone. The hormone actually a lot of things to explain already. Okay, then this is first trimester, and after the first trimester, you notice the human chorionic gonadotropin to be decreasing, and then it's kind of just maintained constant in second and third trimester. So the reason for that is after the first trimester, we only need corpus luteum in the first trimester. Second and third trimester, corpus luteum is useless because we need a lot of androgen and progesterone, and it can only be achieved by something big, right? Something big need to secrete a lot of it. Corpus luteum is just like this small. But we have now, second and third trimester, the placenta is fully developed already. And placenta does the same thing, but better. Placenta secretes estrogen and progesterone. It also secretes. That's why you notice after second trimester, human chorionic gonadotropin decrease, corpus luteum will degenerate because of that, and estrogen and progesterone increase. This is because now estrogen and progesterone is secreted by the placenta instead of corpus luteum. So bigger things, bigger surface area, you can secrete more estrogen and progesterone. Then now, the human chorionic gonadotropin drop, the corpus luteum degenerates. That's why I came across a question that asks why in second semester there's a bit of bleeding. So there are a few factors to it. So one of it is because now the estrogen and progesterone increased by a lot already. The endometrium wall is very thick and fragile. So a bit of friction will cause bleeding. So another reason is, if they want you to relate to human chorionic gonadotropin, is since it's dropped, corpus luteum degenerates, corpus luteum needs to be discharged. Therefore, there will be a bit of bleeding, and those are the corpus luteums remaining. Okay, so second trimester is basically that. And third trimester, we will talk about third trimester here. Okay. You will see third trimester, it peaked, the progesterone and estrogen peaked. So this is because in third trimester, the estrogen increased by a lot, because with the presence of high level estrogen, you will induce birth process, or what we call as parturition. Okay, so second and third trimester is not a lot, it's just first trimester there. So last, I will be talking about the hormones during birth process. Okay, there are 4 different types of hormones. I suggest you guys come up with some very special abbreviation to remember all these hormones. So it's oxytocin, estrogen, progesterone, and prostaglandin. First, we will talk about like at the last week of pregnancy, because at the last week of pregnancy is when the birth process happens, is the very last week. So at the birth process, I said just now, estrogen increased by a lot, induced birth process. Therefore, the last week of pregnancy, the estrogen will peak. So the high level of estrogen actually triggers the oxytocin receptor in the mother and the fetus to produce oxytocin. So it's kind of weird for me because you produce something for yourself to trigger. Okay, so the oxytocin will actually cause contraction in the uterus. Okay, the contraction in the uterus. So that's why, like during pregnancy, even before the mother want to give birth, the stomach will start to hurt already. And then actually the contraction comes in like different series. So maybe each series have like 2 hours, 3 hours gap. After first series of contraction, the mom stomach pain already, and then pass to the next one, get stronger, pass to the next one, get stronger, because oxytocin level is increasing over time. So eventually, this is known as rhythmic labor. Okay, so while this contraction occurs, right, you can imagine yourself like holding like a water bottle like that, and then you can squeeze one, you put one ball here, and then you keep squeezing the end, the ball will start to move towards the other side because you are decreasing the area here, so it will just move to somewhere else. You are like pushing it. So while this oxytocin triggers the contraction, the baby will start to move towards the birth canal. Birth canal is where like the vagina there, the baby come out from there. Okay, the baby will start to move towards the birth canal. And then as it moves, right, like the placenta is barely enough to keep the baby there, as you can see in this program, in this diagram. As the baby starts to move, you are like stretching the placenta and you are like applying pressure onto it. So while this happens, the pressure receptors on the cervix, or you're squeezing the whole area. So the cervix and placenta, there's pressure receptor, and it will sense it and it secretes even more oxytocin. Then the mom is like, oh my God, so painful already. And then you secrete some more oxytocin, it will contract even harder. So the contraction, like as it gets very, very strong, the contraction already, and then at this time, you secrete prostaglandin. The placenta will secrete the prostaglandin. And then, guess what? This prostaglandin causes it to contract even harder, okay? Stronger and more frequently. So you need to keep in mind, this prostaglandin secretes when the baby is very near to the birth canal already. So that's why you need the one last push. So prostaglandin helps to provide the one last push there. Okay, and there are a few positive feedback mechanisms that we need to know during the birth process here. So, like the babies are being pushed towards the birth canal, the mom very pain already. You can see like she's damn stressed there. She have physical and emotional stress. She's like, oh my God, am I gonna live through this? That's the emotional stress there. Okay, then this will actually trigger the brain to secrete even more prostaglandin and oxytocin. This is not because the brain want to torture the mom. This is because the brain is like, okay, she cannot take more of this already. We need to shorten the time and then just fasten the process. That's why prostaglandin and oxytocin is secreted. Then obviously there's more prostaglandin and oxytocin, the contraction will intensify, and then this will allow the baby to move towards the birth canal faster. Then once the baby comes out, right, the pressure is gone already, the mom also relaxed already, the no emotional and physical stress, the prostaglandin and the oxytocin actually stops, the secretion of them is stopped. And once the secretion is stopped, the body senses that, and then the contraction, the labor contraction will stop. So therefore, everything will stop, the mom gets to calm down and happily look at her baby and stuff. Okay, see, prostaglandin acts to contract and expand the uterus to minimize bleeding and maintain the blood volume also. And that's all for my presentation. Oh shit, Roman.
Okay. Job well done to you as well, Pang. So, I bet after all the discussions and all the presentations being shared by our power leaders from KMS just now, are readily understood by everyone in the YouTube streaming, okay? So, because I have to say once again, basically from my perspective as a student that never takes biology, even me also can understand well of what are they trying to say, because the way they explain is really clear. And to everyone's information, they actually made all the PPT slide by their own. Yeah, they only refer to sources and they create their own. It really takes a lot of effort. It took them a lot of effort and time. That's why they came up with some great stuff, with some great masterpieces that is readily understood by everyone. Okay, so I think we are moving into our Q&A sessions in which we have a lot more to answer. Okay, so I'm going to start from the one that we had just now. Okay, so this is a question from Nick. I think this question was supposed to be forwarded to Amira. So it does in autopolyploidy that occurs doubling chromosome occur in meiosis too?
Okay, so as this is actually the W of chromosome, which is replication, it occurs after the meiosis. It does not occur in the meiosis because we can after the meiosis the sterile hybrid will produce. So after the sterile hybrid produce, it will undergoes replication to produce the fertile hybrid.
Okay. Great presentation. Of course, expected. Okay. Oh, another question from Nabila. In transformation, could you explain how come those three conditions happen? Yeah, I reckon you guys mentioned those conditions just now.
Like there will be three conditions, right? One of it is the host site receives the recombinant plasmid. So this happens when the target gene already inserted into the plasmid. So then we'll have recombinant plasmid lah. Then if the target gene is not inserted into the recombinant plasmid, it will form non-recombinant plasmid. And then yang two will be transformed into the host cell. Whereas for the no plasmid one, uh, no plasmid will be transformed into the host cell. That's all. Actually, there's plasmid and then there's host cell. Then there's something called calcium chloride heat shock method. Okay, so during that method, some of the plasmid will go, some of the plasmid won't go.
Okay, crystal clear explanation from Seema. Oh, guys, the making. Pang's first name is Tyrone. Really?
Yeah,
You
my
have an
name is
English name, huh?
Steven.
Oh, because
Uh,
Tyrone, I only know Tyrone Mings from Aston Villa.
no,
Okay,
it's a meme. I know this guy.
okay. Next question. Wow, Pang is so pretty. Pang, I should dub your gender. Are you a boy or girl?
I mean, dude, it's 2020. Dude. You can just say there's more than two. All right.
Oh, there's more than two. Yeah, it's more than currently. We have to be open-minded about this. Okay,
Yes.
another question from Saifullah Aziz. Does target DNA and target gene DNA are the same thing?
Actually, I think there's only target DNA or gene of interest. I've never heard of target gene DNA.
Okay. Well, if Saifullah have anything to add up, just write in the comments below if you seen our reply. Okay. So another question from Nick. The secondary oocyte and the graphene follicle is different.
Yeah, it's different, and the way that the graphene follicle is much larger than the secondary follicle. So sorry, the secondary oxide is found inside the graphene follicle, and it is released during ovulation when the graphene follicle fuses with the wall of the uterus. Yeah.
Okay. So another question from our solid fan, AZ. What is the ploidy, the haploid and diploid thing of primary follicle, secondary follicle, and graphene follicle?
Okay, so for primary follicle, it is still 2N or diploid because it hasn't undergone meiosis completely yet. So at secondary follicle, it becomes N or haploid because it's finished meiosis 1, and the same goes for graphene follicle, diploid as well. Haploid as well.
Yeah, diploid and haploid are the two things that I used to mess up in my Form 4 and Form 5. And when you messed up both of them, and you're gonna lose marks for it. Okay. So Easy agrees with Pang's comment just now. It's a popular question in essay. Okay. So we go for the next one. The question from Alia. Did hydrolytic enzymes and hyaluronidase is the same thing? What is that?
Actually, that is not in our syllabus, but the hyaluronidase thing is it's not an acid that is used to hydrolyze the zona pellucida. It's used to hydrolyze something else, not the zona pellucida. I'm not quite sure what is that. Sorry.
Oh, so it isn't in our syllabus, is it?
No, I at least I haven't heard of it yet until now.
Okay. Biology is really complicated. When it comes to enzyme, I only remember those what you call lipase or those things. And the only intracellular enzyme that I could remember is the oxidoreductase. Yeah, that we study in Form 4. That's the only thing I could remember.
Yeah.
Nicholas Loy, I love you, Pang. Okay, go ahead. Pang, share your contact right below later. Okay, so we have Tan Yen Wen. What will happen to the zygote if the zona pellucida is not hardened after fertilization occur? Else, what is the effect of polyspermy?
Okay. We learned that the polyspermy is actually rare in human back in chapter 7, where we learned that if it's like if it's abnormal number, let's just say it's like 2n, 4n, 5n, the zygote will actually fail to develop. So if the polyspermy occurs, the zygote will just die and no babies will be formed. And if the zona pellucida is not hardened, then polyspermy will occur, and then the zygote will just degenerate. It will not develop further.
Yeah, actually, the consequence is death zygote, but actually there are 2 cases of human polyspermy leading to birth of children have been reported, but it's really rare to success.
Yeah, very rare.
Okay. So how about the effect of the polyspermy?
The effect of polyspermy, the zygote will just degenerate.
Oh, okay.
Yeah.
Okay, so the next question will be the one from Farhana. What cell is divided repeatedly in spermatogenesis process?
Actually, they all undergoes cell division. I'm like from the primordial germ cell all the way to the spermatid and then differentiate into spermatozoa. They all are dividing rapidly. It just depends whether it's mitosis or meiosis only. So actually, I don't know which one you are talking about. Like,
Yeah. In the case of mitosis, if you're talking about mitosis, I'm not sure which one, but if mitosis, the spermatogonia will undergo mitosis. Each of the spermatogonia will undergo mitosis many times until a large amount is produced. Yeah.
So any add-on from Amira and Seema about this? Or else Farhana can make yourself clear later by writing the things that you're referring to, okay? Down in the comment below.
Yeah.
Okay, another one from Easy. HCG's function is to maintain the corpus luteum and first trimester, right? Why does HCG continue to be secreted when corpus luteum is already discharged and replaced with placenta? That's a long question. And of course, a hard one.
I believe it's because placenta it itself secretes some of the HCG also, because chorion initially secretes it because it wants to maintain the corpus luteum there. I believe, okay, I'm not sure about this. I believe that the HCG is also required to a certain degree to maintain the placenta there, to keep other things functioning, because as we all know, there's positive and negative feedback mechanism. Maybe when there's too low level of HCG, it will trigger a negative feedback mechanism on one of the enzyme, one of the hormone there, which will lead to like the baby not being able to bond properly.
So I suppose that will be the full answer for Easy's question. Okay, so let's move on to the next one. May I know the differences between twins and conjoined twins? Because babies, they're babies. I can tell the similarities.
Yeah, so the first difference is in order to form an identical twin, one egg will develop into 2 babies. Oh, sorry, one fertilized egg will split into 2 babies with the exact same genetic material. But if it's for a conjoined twin, it is when the egg fails to, it only partially separates, so it doesn't separate fully. So it will form 2 individuals which are physically connected to each other. Yeah.
Okay.
Okay,
May I know how the
so for
gametes
this,
of twins and
okay,
conjoined twins are formed?
so for this, once the things are fused ready, you form ovum and then zygote, right? And the zygote will actually undergoes repeated mitosis later to form like all those blastula and gastrula, all those things. So while these divisions are occurring, uh, some error will occur, and then it will actually cleave this one cell into two different cells, and then these two cells will continue to undergo division, and therefore will form two babies under one placenta or different placenta. It depends on the condition.
Okay, brilliant answer from Pong. There's probably have a lot of fans, bunch of fans down there. Okay, Tanya Evan again. May I know if the oxytocin play its role in stimulating the uterine contraction if the woman is not going to have a vaginal birth? Oh, I expect this question is going to be really professional. It's going to be really hard.
Okay, I assume you asked if the person go for like surgery and give birth. So
Yeah.
yes, you will still have the oxytocin, and it does play a role because oxytocin actually it signals the mother that she needs to give birth already, like it is time. Okay, so you need the oxytocin to start the labor, rhythmic labor, and then after that the mom will be brought towards the ER, and then they will cut open her tummy and then they take the baby out. So the oxytocin actually plays a role to let us know that she's ready to give birth already.
Yeah,
Okay,
she's ready to deliver, right?
Yeah. I mean,
Yeah,
like,
Nicholas.
okay, it
Yeah.
doesn't it doesn't do everything I just explained. Like when the baby moves towards the birth canal, it will secrete more, but the initial one will happen because it occurs without the baby moving. It occurs because the oxytocin receptor secretes it. So after that, since you remove from the stomach through surgery, it won't move that close towards the birth canal. Prostaglandin and all those won't be secreted. So yeah.
oh, so I thought this question was trying to say that maybe the roles of oxytocin when the woman is not giving birth. So oxytocin actually has other functions as well, even when the person is not pregnant. So for females specifically, oxytocin will help them to reach orgasm during copulation or sexual activity. So it will help them to stimulate the uterine contraction so that they can reach orgasm. Yeah.
Okay. So that means basically oxytocin is not just functioning when the person is giving birth, right?
Yes.
Okay. Pauline Andrew. Wow, thank you, Pang, for the clear explanation. Really appreciate it. Pang, why
Welcome.
did they keep on mentioning you but not Amira, Seema, and Nicholas? Tell me why.
Dude, I don't know, dude.
It's not fair. It's not fair. It's not fair.
KMS top student is Tang.
Nor Anis with a cat character. Is it normal bleeding too much after first trimester or it will cause the abortion? It's about delivery.
You should only have spotting after the first trimester because it's just a bit of friction towards the endometrium wall, or is the corpus luteum degenerating. But if it's continuous bleeding, then the person should probably see a doctor because it will lead to abortion.
Oh,
Because
okay.
if a heavy bleeding occur, it can also cause the miscarriage.
Yeah.
Yeah, basically it is really dangerous to both
Yes.
the woman the pregnant woman and the baby as well.
Yeah.
Okay, Daniels, Dandelion, great job, leaders. Okay, it is a appreciation to four of you. Go, Amira. Amira is going. Happy go lucky. Get ready to be chased in KMS later. Oh, I suppose it's your classmate. I suppose either classmates or roommate.
My friend, yeah, but still.
Okay, what caused premature baby?
Okay, so when you are pregnant and if you take stuff like drugs, medication, or any form of medication that will cause hormone imbalance, any form that cause a peak in estrogen. And as I said just now, estrogen actually actually like induce birth, right? Like if you take anything that causes you to release excessive amount of estrogen, your body might like get confused and be like, oh, is it time yet? All right, let's just start the contraction. So that is why premature birth happens. That's
So,
one of
but
the factors. Yeah.
does premature baby dangerous to the woman pregnant, to the pregnant woman?
It's more dangerous towards the baby because all the process of development haven't been completed yet. So when they come out, maybe their lung is not fully developed or their heart is not fully developed.
So that's hard for them to live, right?
Yeah.
Oh, okay, Saifullah, my bad. Okay, it's not your fault. It's not your fault. We are here to clarify ourselves. We are here to answer a question. It's nothing to do with your fault, okay? Does target DNA and gene of interest are the same thing? So he corrected himself in the question above.
Yeah. Okay.
So that's it, right?
That's it. That's it.
A brief and precise answer, I suppose. Nurul Liana, okay, thank you. Okay, appreciation to our 4 biology leaders again. In birth process, which hormone comes first? Oh, a tricky question, I suppose.
In birth process, the estrogen will come first because estrogen need to like induce the birth process. Therefore, it's technically the first hormone in the birth process. Then only followed by oxytocin, prostaglandin.
So everyone agree with that answer, right?
Yeah, but to be honest, most of the enzyme, most of the hormones, they exist around the same time. So yeah.
Okay.
Yeah.
Like almost simultaneously, because it's important. Every hormone is important, right?
Yeah.
Lai Yifei, may I know how does the twin form? It's something to do with the sperm and ovum, I suppose.
Yeah. If the zona pellucida didn't harden in time, it might one extra sperm might like get in there, like squeeze into the ovum, and then they will like form a twin, or like just now we say during the process of like implantation, before the process, the sperm, the cell might split into two, and then therefore twin is formed. So yeah, there's two conditions there.
Okay. So that means one of the situation would be like maybe the sperm arrives at the last minute, right?
Yeah.
Okay. People who came late will not be entertained. Alice, actually, I don't know why I'm here, but thank you. Yeah, welcome, Alice. You can be here for all the time. From Monday to Thursday, you can be here and you're going to see my face all the time. Sorry for that. Okay, so Hidayati, in Aloe Polyploidy fetal, why polyploidy occur and in where process it occur?
Okay, so polyploidy actually occur because of non-disjunction in the chromosome. Because the polyploidy is under the changes in the chromosome structure, right? Okay, so chromosome number, I'm sorry. And then where process it will occur, yes. The Aloe polyploidy usually occur in plants, but not in animals.
Okay, we've got a really tricky question from Junwei. Which day range is the best to do sexual intercourse and high chance to get pregnant? Okay, we're going to judge it from a very scientific and professional perspective. The graph has been sent to Pang Pang. Did you receive anything? Maybe he could share screen. Sorry for the inconvenience. Oh, I guess our polyers are really excellent. They can just answer straight away. They don't have to refer to any graph. Pang.
Okay. I was opening. Okay. So you need to have the intercourse at like day 14. Day 14 is the ovulation, ma. You need to give it like about 1 to about 4 days, 1 to 3 days for it to reach your oviduct. That is where fertilization occurs. So you need to have intercourse like maybe like 4 days, so maybe day 10. So the first day is when you start menstrual cycle. The first day is when she bleeds, and then when she stops, you count out like 10 days like that. You maybe you like have intercourse with her or something.
Okay. So is Junwei your friend?
He was my old classmate.
Oh, I thought he's from KMS. Okay.
Yeah.
Alice. Oh wait, I'm here for Seema. Yeah, finally Seema has got a fan with her. Not only Pang. Amira has also got her fans as well. Only Nicholas doesn't have any fan currently. Nicholas, keep it up.
Space problem.
Hey, no. If you think you have face problem, I'm even worse. Okay. Oh yeah, what happened to the baby that was born before the due time? I think it's something to do with premature baby, like just now.
Yeah, basically the organ is not fully developed and the baby might die.
Yeah.
But yeah, usually the baby is put in the incubator where like they can have a good temperature and then with enough nutrients so that they can try to grow normally.
So it's a new technology, huh?
Yeah.
Yeah.
Okay. People are getting smarter day by day. We've got a question from Angel. Angel, yeah. For chapter 8, when we cut the gene of interest and produce sticky and blunt end, at the same time we break the phosphodiester bond for these two cases. When will we break the hydrogen bond? Thank you. Two questions? Quite long.
No, it's actually when we cut the gene of interest, we will produce either sticky or blunt end. It's like or not and. And like phosphodiester bond is like the bonds between the nucleotide. And then this hydrogen bond is the bond between the nitrogenous group. Okay, so when you cut it, both will form. Okay. Sorry.
Okay, so that's your explanation from Seema, right? Is it completed? Okay. So a third question or the fourth question from Daniel Wen. No, it's not a question currently. Thank you so much for the clear explanation. By the way, the question I asked just now is about the Caesarean birth. What is Caesarean birth? Is so new to me.
I believe Caesarean birth is the one where you, when Tang explained, where you do an operation on the abdomen there to remove the baby.
Yeah, is to surgically deliver the baby.
Oh, okay. From Lai Fei, thank you so much, Brocon. Thank you for all. Salute. Yeah, salute. Okay. Oh, Nicholas, you've got a team of fans, not only one.
Oh, you're your first friend.
Among 5 of us, I'm the only one that doesn't have any fans, but it's nothing to me because I'm not presenting. Okay.
We need Team John in the chat.
No.
Team John in the chat.
Angel. After fertilization, what happened to antipodal and embryo sac? Oh yeah, I could remember this question from Form 5. Similar to Form 5.
Let's go, Nicholas.
Nicholas, let's get it.
Damn,
Yeah. No, after fertilization. Okay, so the antipodal sac, so the embryo sac, which is basically the ovum part, will develop into the flesh of the fruit, together with everything inside. Yeah. The flesh of the fruit.
The flesh of the fruit. Okay. I hope it makes it clear for you, Angel. Angel. Sue she's got a question. What is blunt end for?
Blunt is like other recombinant DNA technology. So in the slide, I only talk about the recombinant DNA technology, which causes insulin production. Recombinant DNA technology can cause a lot of things. So blunt end is for the lot of.
Okay, thank
Oh
you.
wait, sorry, I would like to make a correction. The embryo side develops into the seed. Yeah, not the flesh of the fruit. Yeah.
Oh yeah, the question from Angel.
I just checked my book. Yeah.
Okay, it's okay. Everyone made a mistake, but overall you're great.
You're the best, bro.
Everyone is the best. Okay, even our audience are the best. Why we need to place premature in incubator? I think Nick, you mentioned something about incubator just now.
Oh yeah, it's because the baby is not well developed yet. It's still premature, like 6-month baby. So it has to have like normal temperature, like the same temperature, same conditions as inside the uterus lah. So basically, that's what the incubator is for.
Okay, from Kang Jenjack. Good job, you all. Oh, I would like to make a brief introduction about who is Kang Jenjack. Kang Jenjack is one of the exco kebajikan for KMS. Yeah, thank you, Kang. Saifullah Aziz, thank you so much, Nicholas, Pang, Amira, Seema, and you. Yes, finally I'm being mentioned.
Team John, let's go.
Yeah. Okay, so for the moment being, this will be the last question. Oh yeah, we've got some more. It's Nick again. Yeah, I could recognize her. Does the target DNA can be taken from? Oh, she's going to do something about surgeon.
I think you must take from human pancreatic cell because human pancreatic cell is the one that produces insulin, and then what human pancreatic cell will have mRNA. I think so. Not sure. I think so.
Okay. You got this. I suppose this will be an emoji. Aisha, what is the importance of marker gene, and is it connected with antibiotics? By the way, thank you guys. Welcome.
The marker gene. Now I when you want to differentiate the bacteria with plasmid and without plasmid, the one without. Wait, let me rephrase it. Okay, so when you don't have plasmid, you won't have the marker gene. Okay, so remember when I said transformation got three consequences, right? One of it is the whole cell won't have plasmid, kan? So the one without if you don't have marker gene, you cannot differentiate it. So that's the importance of the marker. And antibiotics, like different plasmid will have different antibiotic resistant gene. For PUC18, it's antigen antibiotic.
Okay, thank you. Thank you, Seema. I love you, Pan. Okay, Nicholas, we've got your point for a lot of times. You've mentioned it a lot of times. Got enough. Okay, we got enough for Pang. Maybe you can mention for Amira, Seema, Nicholas. Okay. Nicholas. Oh, he even supports Nicholas Loy just now. So Pang is the best. Okay. Okay. No, no
There can
bugging.
only be one best, dude. Pick your side.
Okay, Nicholas Insta. Show me. Okay, give me your Insta right away. Okay, welcome, Lisa. Pang, create a fan club. Yeah. Oh, okay. Planning. I bet Pang is planning. Okay, welcome, Umara. Okay, he wants Pang Insta. Just now is Nicholas Insta, now it's Pang Insta. Not fair. Thank you so much, Amira, Seema, Nicholas, and Pang. Yeah, they did a really good job, of course. Okay, now guys, I'm going to share a link that are going to direct you all to this Google Form link, which I'm going to share to you all. So hopefully you guys can help us to fill it up. It's about the feedback and response about our content of presentation today. So it's already in the comments below if you guys are free. I hope you guys can fill it for us. Okay, another question. Power leader from KMJ is over here. Oh, she is a power leader from KMJ. Good job, guys. Enjoy revising tonight. Thank you so much, John. Oh, she's just trying to give me the feeling of getting name mentioned. Okay, but by the way, I'm satisfied. Thank you. Yeah, I purposely show this John Insta. Okay, I got your point. I'm going to share later. Thank you for the explanation. Okay, Nicole Jade, welcome. Thank you all parties for a great presentation and clear presentation. Okay, welcome. Thank you all, welcome. Okay, it has passed 10 o'clock currently, but I wish to say something again. As the president of Power Program in KMS, I wish to bid my appreciation and gratitude to all of our leaders involved into this program, which are Amira, Seema, Nicholas, and Pang. They did a really well job because they really prepare well for this. They prepare for a lot of times. They put in a lot of efforts. Of course, the outcome was really excellent just now. And for you guys to like present your feedback, you can just fill in the Google form that I've presented to everyone just now. This one, and you can find in the comment there. Okay, Serena, thanks you. Okay, welcome. Welcome, Nurul. Okay, welcome, Hidayati. So I bet that's all for our presentation today. So we're going to continue with physics and accounting for tomorrow. at 8 o'clock as usual, and we're going to have a different batch of power leaders. So hopefully you guys can join us as well, okay? Because today's attendance is really excellent to me, around 300 people. And I wish there are more than 300 people starting from tomorrow, okay? So I need everyone's support from other colleges as well as KMS. Thank you for the clear explanation. Is it still going to be Pang tomorrow? Pang, they need you for physics. Oh, sorry, Pang is module 3. Pang doesn't have any physics. The same
I
goes
don't
with
take
account.
physics, bruh.
But if you guys are really demanding for Pang to be here, I can bring him in every day. He's going to take over my place as the moderator. Can. I'm very open-minded. Okay, so I think that's all for today. So we're gonna end our presentation in a count of 10. So everyone, thanks for your participation. You are free to leave.
Yeah.
Thank you.
Okay.
Thanks,
Thank
guys.
you.
Thanks for
Thank you.
watching.
Thank you for watching.
I'll drop
Subscribe to the
Nicholas'
channel,
phone
leave
number
a
in the
like.
chat later.
And
I think
yeah.
I've already exceeded 100 subscribers.
Okay. Yeah. Have fun studying.
How about
No, dude, why are you doing this? Leave me alone, man. This is cyberbullying,
chemistry?
dude.
Can, Pang, join. Thank you. Okay, so that's for all. Bye bye, guys. Good night.
Bye bye.
Bye.